Good day, my dear young intellectuals. How was your asynchronous learning so far? I hope that you are learning something from the different resources and materials that I provided to you. In this video, we are going to focus on experimental variables, which is still under Unit 1, entitled Doing Scientific Investigations. In this lesson, your objective is to differentiate independent, dependent, and control variables. So are you ready to learn? Let's start. So our previous topic is about the steps of scientific method. Now let us go back to our previous experiment about fertilizers. So before you conduct your experiment, you observe that a fertilizer can make the plant grow bigger and faster. That's why your question in mind will be which type of fertilizer works the best. And based from your researches conducted, you found out that there are 3 available fertilizers, but you don't know yet which one is the best. That's why you formulated a hypothesis or a possible answer to your question or an educated guess that plants grown with fertilizer A will grow the fastest. So to make sure that this hypothesis is correct or not, you need to conduct an experiment. Based from your experiment, your results showed that plants with fertilizer A is the best fertilizer or it's the best result. Therefore, your conclusion is your hypothesis was proven correct. So you're now going to accept this hypothesis in which the fertilizer A is the best fertilizer among the 3. Now, let us focus on the hypothesis. What is hypothesis again? Hypothesis is an educated guess which is also the possible solution to your problem, and it should be tested through an experiment to identify whether we accept or disprove it. So let us take this as our hypothesis. If I add fertilizer A in an orchid, then the growth of the orchid will increase. So as you can see, the format of hypothesis is it starts with if followed by then. But you may wonder, what is the connection of hypothesis to our topic for today? Well, as we test our hypothesis through an experiment, we should identify our variables. So what is a variable? A variable is any factor that is being tested in an experiment. So in short, these are the actors and actresses that plays a very important role in your experiment. So before you conduct your experiment, you should be able to identify your variables first. And there are 3 kinds of variables in an experiment. First one, independent variable. The next one, dependent variable. And the third one is the control variable. So, let us first describe what independent variable is. Independent variable are factors that we change or manipulate during an experiment. So if you wish to identify what will be your independent variable in your experiment, you should ask the following question. What factors do I want to change or manipulate? So in identifying independent variable, you should focus on the things that you want to change or you want to manipulate. So based from our situation in which we are going to identify which fertilizer is the best for the plant growth, obviously the factor that we want to change or manipulate or the thing that we want to use during the experiment is the type of fertilizer. whether it will be fertilizer A, fertilizer B, or fertilizer Z. Next, dependent variable. Dependent variable, on the other hand, are the factors that you want to observe or measure. So take note, dependent variable, the things that you want to observe or measure during an experiment. So if you want to identify your dependent variable, you should ask the following question. What factors do I want to observe or measure? So going back to our situation, we want to measure the plant. So therefore, our dependent variable is plant height, because plant height is the factor that we want to observe or measure as we apply the independent variable or in our case, the type of fertilizer. So after applying the fertilizer, what are we going to observe or measure afterwards? Obviously, the plant height. And the last type of variable is the control variable. These are the factors that should be kept constant or the same. It should not affect the result other than the independent variable. That's why it is called as control variables, because these are the factors that you should control and should not affect the result of the experiment other than the independent variable. Because in an experiment, we want to make sure that it is only the independent variable will affect the result of the experiment. So in short, control variable tells us that one for all for one should be applied or should be practiced. Whatever the factors that we apply to one setup should be applied for the others as well. So if you want to identify your control variables, you need to ask this question. What factors will I need to keep constant or the same? So in our experiment, what could be our possible control variables? So it can be the type of soil, the species of the plant, amount of fertilizer, exposure to sunlight. So in short, control variables can be more than one, and these are the factors that we want to remain the same for all experimental setups. We need to make sure that it is only the type of fertilizer that will affect the plant height and not the type of soil, the species of plant, the amount of fertilizer, and the exposure to sunlight. So therefore, these are the factors that should be the same for all. So if we apply sand to an experimental setup plant, we should also apply sand to others as well. If our plant is orchid, the other plant should be orchid also. If we apply 50 grams of fertilizer in one plant, we should also apply the same amount of fertilizer for all. And if this plant will be exposed to sunlight for about 8 hours, let the others be exposed to sunlight as well for about 8 hours. So therefore, control variables are the factors that should be kept the same. Now, let us now see the connection of experimental variables in our hypothesis. So as you can see, in our hypothesis, we can observe now the independent variable, which is the fertilizer A. And what are we going to observe if we add fertilizer A in an orchid? Obviously, our dependent variable is the growth of the plant, because the growth of the plant is the factor that we want to observe after applying the independent variable, in our case that is the fertilizer A. Another example, if I use 75 grams of sugar in a bread dough, then the bread will rise. So which one is the independent variable and which one is the dependent variable in this hypothesis? Okay, so our independent variable is 75 grams of sugar because we are going to manipulate or change the amount of sugar. That because the sugar or the amount of sugar is the factor that we want to use. Whereas the factor that we want to observe after adding 75 grams of sugar will be the dependent variable, which is the height of the bread as it being added by the amount of sugar. Now, it is very important to remember that independent variable influences the change in the dependent variable. I will repeat, the independent variable or the factors that we want to change or manipulate influences the change in the dependent variable or the factors that we want to observe or measure. Other than that, the control variables should be kept the same or constant. So let us have another example. So the independent variable is the one thing that you change. It is limited to only one during an experiment. So in this experiment, our independent variable or the factor that we are manipulating or being changed is the liquid used to water each plant. So in plant A, the liquid that we use is a soft drink. In letter B, we use a juice. And in letter C, we used water. So in identifying independent variable, it is important to note that independent variable are the factors that we change or manipulate, that we used during an experiment. The dependent variable, the change that happens because of the independent variable. So after applying the different liquid, what are we going to measure or observe? Obviously, the height or the health of the plant. And the control variable is everything you want to remain constant and unchanging. So other than the liquid used in watering each plant, the factors that we want to remain constant or the same is the type of plant use, the pot size, the amount of liquid, the soil type, etc. Okay, so let us have some exercises. So I want you to identify the independent, dependent, and controlled variables to the following experimental setups. So number 1, different rose bushes are grown in a greenhouse for 2 months. The number of flowers on each bush is counted at the end of the experiment. So based on the situation, what is our independent variable? Okay, so our independent variable is the different rose bushes because this is the factor that we are manipulating. What are the factors or what is the factor that we want to observe? Or what is our dependent variable? So the number of flowers is our dependent variable. And what are the controlled variables? The kind of flower, greenhouse, temperature, type of soil. Okay, so let us have another one. You water 3 sunflower plants with salt water. Each plant receives a different concentration of salt solutions. After 2-week experiment, the height is measured. So what is our independent variable? Obviously, the different concentration of salt solutions. What is the factor that we want to observe? The plant height. So it is stated here very clearly. And what are the factors that we want to remain the same? So the amount of salt solution, because we are just going to test which concentration is the best and not the amount. the kind of flower, so it should be sunflower, the type of soil, temperature, and the location of the setup. And then for our last example, you want to determine the effectiveness of your invented vaccine against coronavirus. You will look for 10 participants who will receive your experimental vaccine. So what is the factor that we want to identify or we want to measure or manipulate? Obviously, that is the vaccine. What is the factor that we want to observe? The immunity against COVID-19. And what is the factors that we want to remain the same or constant for all? The type of participants. So if our participants are humans, so all of them should be humans and we should not include other animals. The amount of vaccine given. The number of participants. So it is stated here that we are going to look for 10 participants. And the time of experiment. Okay? So I hope that you are learning something about this lesson. Let us now summarize the lesson. So, variables are factors that is being tested in an experiment. And there are 3 types of variables. Independent variables are the factors that you change or manipulate, while the dependent variables are the factors that you measure in response to the independent variable. And the last type of variable is the control variables, or the factors that should remain the same or does not influence the result of the experiment. So I hope that you are learning something about this lesson. Thank you for listening and see you on the next lesson.